Now
Yes.
it should be okay?
This is perfect. Thank you.
Okay, thank you. Yes, okay. Actually, I can cut this talk very short, which is titled, Can Archaeology Save the Sea Turtles? And the answer to that is no, it cannot save the sea turtles. So here you have it. But it's an interesting idea, isn't it? In this day and age when we are very much embedded in the Anthropocene. The Anthropocene is well underway, and although until recently it was just the domain of geologists, it is a term coined by geologists. We understand more and more increasingly, as they said in that movie, Don't Look Up, everything is connected, right? So although, as the name suggests, in the Anthropocene, it's the era of the humans are on top of everything. So the implication being humans have mastered everything and they have impact on everything we understand more and more. This is all connected and it's complex and it's more nuanced. And so I'm a zooarchaeologist, so I'm a bioarchaeologist, and I think bioarchaeology has something to add to this. And how is that? So, and that is, of course, depends on the fact that it's based on the fact or the idea that it's all connected. So there is no real difference between nature and culture. This dualism is a very recent dualism in our understanding of ourselves, our history, our place in the world. So then, and I was lucky enough, yes, I was never an archaeology student in the department, but I was going to excavations at Canan, which you all know this site really, really well, that Maria Harriet Gates and Charles Gates and other people, students of Bilkent, have been excavating between in the 90s and 2000s, early 2000s. And this is a site at in the coast here of the Mediterranean. It's a multi-period site, of course, like many of our sites in the Near East. And it has an amazing, amazing zooarchaeological record where I learned zooarchaeology. So today I want to focus on my narrative on this site and on the sea turtles of this site, which we recently did some publications on and we still are working on, because this narrative, I think, explains the idea what bioarchaeology can do for us and our place in the Anthropocene from my point of view. Okay, here I am, and this is also Salima Ikram's work, Professor Ikram from University of Cairo, among others. So Canan, our multi-period site, starts from the well, earlier, but early Bronze Age, and it has time periods, phases until the medieval period. So interestingly, there are, I mean, it's on the coast and on the coast of the Mediterranean. So there are fish, there are shells, and there are a lot of domesticated animals. Most of the taxa here are domesticated animals. And at some point, there are also, in each layer, a few remains of sea turtles, right? But in the Middle Iron Age and until the end of the Late Iron Age, there are more sea turtles. So every context has remains of sea turtles. Well, quite interestingly, this period, by and large, coincides partially with the presence of Assyrians, Neo-Assyrians in the area. And you have this wonderful relief from Khorsabad, the famous Khorsabad relief, which depicts these, well, some sea turtles. So, well, I met these sea turtle remains ever since I started excavating at, working with the zooarchaeology of Kinetheuk in 1999. And at the time, we didn't know much about these bones. And we knew that there are three different species in the Mediterranean as sea turtles. These are the true sea turtles, and then there's the Nile soft-shell turtle that roams in the sea, but it's a different lineage. But we didn't know what they were really. So then I graduated from Bilkent and I moved on to American University of Beirut, and I got involved in different excavations along the Lebanese coast. And every excavation pretty much that I've ever since then been to on the coast would have sea turtle remains. This is one of them. This is an early Bronze Age site, very close to Byblos. Lots of sea turtle remains here, which we could identify as sea turtles. This is one of them. This is an exceptional example. And then I would go several years later to Professor Yaşar Ersoy's excavations at Klazamanai on the Aegean coast. And here they are again in how we usually find them in very fragmented form. And this is the reason why we could not really identify them to species. And this is where you always have to begin. You have to know your species, right? But there were no tools, no literature, no anatomical atlas, nothing. And this is how they would appear. So what we started doing was, of course, recording them anyway, the zooarchaeological manner. Are they shells of the turtles? Are they body parts? These are the flipper bones that are heavily fragmented. And so we decided, okay, what do? We need to know the species. So together with my student, Francis Koistra, we went to a bunch of natural history museum collections, and we looked at the morphologies of turtles to identify our very fragmented remains to species. And it worked. It worked on not the shells, but on the fragmented other body parts like flippers and especially the flippers and the backbones. So we were able to do that just like we would do with other zooarchaeological records. There were also osteometric, so biometric shape analysis we did, and we could tell the difference between Caretta, the famous Caretta, and the Chelonia midas, the green turtle. Well, all of a sudden, also what we saw was something super interesting. Turtles grow exponentially. So not like us allometrically, but they grow exponentially, which means that if you have one body part, even like this, you can actually reconstruct the size. And then size means age in these animals, right? So we noticed something really interesting, that when we compared our size reconstructions of green turtles to museum specimens, all our archaeological specimens were large, much larger than any specimens that we would find in museums like the Smithsonian or in Brussels. So that was really interesting, and something we also discovered along the way as we got to know better the anatomy of the turtle is that we were finding the remains of male turtles. And this is to do with the little phalanx here. Well, in the males, this phalanx, this finger, it's really, really big so that they can mount on the females. This coupling happens in the sea. And in the females, they're very small. And at Kinet, we found a lot of these, which indicates that people were not collecting the females, but they were actually hunting these males. So we had a better insight into hunting. We also got, at all of these sites where we found turtles, better insight into the processing. For instance, we understood that based on the butchery marks, based on the presence of the body parts, that they've brought all the turtles into the sites. They processed them there in a rather systematic way, actually how they would do with the domesticated turtles. And then we also collected other evidence from other sites from the literature. And we were able to map that these sea turtles, they were there since the Neolithic. And yeah, so almost every coastal site had a few or more sea turtle remains. They were ubiquitous, but not awfully numerous. So the Canan case was quite an exception, we found out, right? And then this made us, my slide thing doesn't work for some reason. What happened here? Okay. Yes, sorry, now it works. I think the computer is being slow. So this made us think that by and large, turtles were a taboo meat, perhaps. So there were exceptions sometimes that they were hunted, they were captured in the sea, but mostly they were avoided. Taboo is a big word, avoided. But at the same time, of course, what was happening is the coastal geomorphology was changing. So when we were finding the turtles on the coastal sites, the coast of the Mediterranean looked entirely different than now. And for instance, at places where we have, this is Patara, where we have turtle nesting and feeding, this beach wasn't there at all when the Iron Age site of Patara was there, where there are no sea turtles. There are, there's one or two. So the sea turtles were there, but they weren't necessarily nesting, perhaps. So that's one of the things that we're working with now. But how do we find out, right? This is the zooarchaeological study that I described. How do we find out more? Well, recent advances in bioarchaeology help us investigate the matter further. This is my PhD student who works on the sea turtle remains from these sites, including Canan, in a larger European Union training program. And now we are able to do much more. For instance, these are the shell parts, turtle shells that we as zooarchaeologists using non-destructive methods, only anatomical methods, we cannot identify to species, you're right? But in the bones, there's collagen, and the collagen in each different species in the world, the collagen has a different sequence. And this sequence we now can read through protein fingerprinting based on their relationships. And we also have done this for the sea turtles. So when we look at the peptide markers for each living sea turtle, there's a different marker. And when we're in the lab, in an isotope lab, we extract the collagen from the sea turtle bones and look at their protein sequence. Even the tiny and fragmented bones or shells of sea turtles, if they have enough collagen, and you really need extremely small parts, now we can identify them to a species. So all of a sudden our picture of the distribution, the amount of different species relative taxonomic composition changed drastically. And one of the other things that we are now doing is we're looking at foraging ecology. So the trophic chain of the sea and of the terrestrial world changes drastically. It has changed drastically. That's one of the impacts of the Anthropocene. And that happens when the environment degrades. One of the species that is food is removed from the environment. And one of the problems with the sea turtles today is actually that we're missing there. We have removed most of the seagrass, and I'm afraid to say some of the removal was done for tourism. Tourists don't like the seagrass. They want the beach, they want the sand, so they were deliberately cut in all over the world. But this is essential food for, essential ecosystem for a lot of animals. And by using stable isotopes of the bones, we can see what the sea turtles used to eat, right? Whether it's seagrass or different types of seagrass, and even looking at the modern studies this is the one in the Mediterranean, by looking at the isotopes, you can track them. So, of course, we cannot track archaeological sea turtles, right? But because the stable isotopic composition of the seawater in different places in the Mediterranean, it's different. This goes into the bones of the sea turtles, and then it stays there for millennia. So in the archaeological sea turtles, we can extract this information. It's nitrogen, carbon, and sulfur, and compare them to the modern situation. And we're doing this right now. And what we're finding out is quite amazing because of this partitioning, isotopic partitioning of the Mediterranean Sea. We can track quite literally the Mediterranean sea turtles. We can tell whether the certain feeding areas and nesting areas that we have today existed in the past or not. So, and what we're also doing is, remember, the question was, okay, so we have few sea turtles. What's going on? Is it the environment? Is it human choice? Well, there's an independent source, that's the genetics. So in addition to that, what we're doing is we're looking at the ancient DNA from the sea turtles. This is also very difficult work to see how large and how diverse and how old these populations were. So now to the excavations. I think this is Kinet. We have done really, really well. Maria Ariette and Charles Gates, they have done amazing excavations. So we're now using, on top of traditional zooarchaeology, these biomolecular methods to understand all sorts of things about human-animal relationships. I gave you the example in sea turtles, and it's important to use these keystone species to understand the Anthropocene better, to understand the historical ecology of human-animal interactions better. But it's also important to focus on these keystone species for the moment and to go the extra mile because the public, it speaks to the public and this public will speak back to you. So that's a great connection between archaeology, anthropology, and ecology and the society. This is, our work was featured in the Atlas magazine in Turkish, and it was also covered by several international newspapers. But that was the whole point, that we focus on sea turtles because the Anthropocene is not just about foraging and the humans exploiting the environment. It's also about all the other human behavior. Okay, so this is my highlight. I know I'm over time. My last slide, hopefully, that is what we can do with bioarchaeology is cool, and it takes a lot of work, and it's advancing really rapidly. On the one hand, it's also a lot of work, a lot of time, but because it's archaeology, it also gives us all this other information, right? The human information, along with the animal or plant or whatever environmental information. So that's important to focus because, as Nicole and Alison put it perfectly in their recent article, what we have to offer is models from the past. But these are not techno fixes, right? So this is how they were doing in the past, so let's do that. No, it's actually a lot about how telling the story of humans' resilience, societal resilience, coping mechanisms, and failures as well, and increasing by this, using this information, increasing the environmental stewardship, right? Environmental awareness, ecological literacy of societies, starting with archaeologists ourselves. And that's the end of my presentation.
Thank you very much.